Here we are at number 9. Actually, we're at page, as you can see here, 83. And we're looking at number 9. We're looking at the mitochondrion. And the mitochondrion is an interesting organelle in that it functions more or less as a captured bacterium, but it's a bacterium that actually is aerobic. Right, so you actually see two membranes. There's the membrane that belongs strictly to the eukaryotic cell. That's this one right here. And then within this membrane is a somewhat, how shall I say, folded little cell, you could say. There's a little area here, a little moat between the two, or a cistern between the two membranes. And then with this membrane, we actually have whole series of enzymes for the purpose of oxidative phosphorylation, I should say. That is to say that we're going to be changing ADP into ATP through these membranes. It's linked to the Krebs cycle, which exists inside this unit as well. So the Krebs cycle is inside here. The enzymes that participate in the Krebs cycle, so that when we metabolize carbohydrates, most of our energy from carbohydrates comes from within the mitochondria. So for 1 molecule of glucose, we're going to get 38 molecules of ATP, high energy currency. Only 2 of those will come from outside the mitochondria as a part of the anaerobic part of glucose metabolism. So aerobic behavior is predicated on this little unit. Fitness, really, I would suggest to you that the size of your biceps has, how shall I say, very little to do with fitness compared to the concentration of mitochondria in your heart and in those very muscles that you're working out with. That it's an issue of, can you get ATP? Can you endure? Can you come back? Can you keep going? That's what endurance is. And so fitness is that. And the heart especially is a place, over here we're talking about the mitochondria here. It says the density of mitochondria in a particular cell reflects that the cell's energy requirements and the mitochondria's general cluster where the action is. So basically, in somebody who's physically active, these individuals will have muscles and then also hearts, skeletal muscle and heart muscle that is rich in mitochondria. One of the most profound things that you can do is exercise. And with a good sleep cycle and good nutrients combined with exercise, it's possible to actually profoundly change the human body in ways that could not otherwise be done. So probably the biggest change you could actually bring about is through good exercise, good sleep, good nutrition. The exercise, by the way, should be, how shall I say, joyful. That if you do something that is difficult or traumatic, you're going to just cause an abundance of cortisol to be released, and that's going to actually prevent the growth hormones, growth factors from the liver from doing their job. This is where we also experience aerobic respiration. This is why we have oxygen. So the final electron acceptor, the final thing that is the result, we're actually able, unlike our machines, we're actually able to burn our fuel all the way to completion. This is a very big deal, to actually burn your fuel 100% all the way to completion, which means you start with glucose and you end with CO2 and water. So oxygen, grabbing the hydrogen from metabolism, is what causes what we call the waters of metabolism, and that's on the far side of oxidative phosphorylation. So we'll be, in Chapter 24, we'll have a very intimate examination of that. Also, another detail here is that the mitochondria has its own bacterial DNA, and it has bacterial ribosomes, which matters. It plays a role in therapy in which antibiotics are used. In some cases, the antibiotics are targeting bacterial ribosomes to prevent them from functioning well. And of course, they will automatically target our own mitochondria. So you could get rather tired when you're under the influence of a antibiotic treatment episode. So it's something to be considered. Maybe we want to make antibiotics function in slightly different ways, more precisely to the bacteria, less to our mitochondria. Okay, so with that in mind, I'm going to turn to another important feature of this chapter, which is the cell cycle, the life of the cell. So let's meditate on that for a moment. On page 97 here, we have the cell cycle, and after it finishes replication from the mother cell, the daughter cell becomes available. If it's not intended to replicate again, we call this G0. That means it's not going to get to this restriction point here, which is the signal that replication is going to become a happening thing. Okay, so we have this special restriction point here, and once that level of growth is arrived at, if it is the type of cell that's destined to replicate a number of times, then in fact it will go into this S phase. Now down here we have something called mitosis, the various phases of mitosis, and up here we have the normal life of the cell, which is referred to as interphase, and together they form the cell cycle of life. And up here is where we have DNA replicated. DNA replication is very important. We have to have twice as much DNA so that the resulting cell, two cells that form, each can get their 46 chromosomes. 46 times 2 is 92, and so that means we're going to have 92 molecules instead of 46, and it's going to happen right here. There's also going to be other things, centrioles that get replicated, so things that have to do with the mitotic event, which I call dosage regulation. That event is going to be predicated on the replication of DNA and the centrioles right here in the S phase. It's S for synthesis. Then we go through more growth. Once we have the replication of the DNA and the centrioles, then we need to have twice as many mitochondria and twice as much endoplasmic reticulum and lots more of everything so that we have enough to divide between two daughter cells. Okay, so this is sort of like pregnancy over here. It's like we're growing enough to create another individual. Okay, and then we proceed with the various stages where you have condensation in prophase, the individuation of the DNA molecules, highly condensed so that they can be moved around properly and dosed correctly. We have metaphase, which means we're going to have an equatorial plane in which the division is accomplished, and there's going to be poles toward which, here's the metaphase right here. So we have the poles toward which half of the molecules can be drawn, and we have to replicate it perfectly so that all 46 are replicated and that there's a chromosome pair of number 1, chromosome pair number 2 are going to each cell. So this is a very organized event, and I call it dosage regulation. We get anaphase and then telophase, and we have the ultimate end of cell division right there. Here is a huge polymerase that's replicating the DNA so that we can have two molecules of DNA. These are actually very huge proteins. They wouldn't even fit on the page if we were to see them in actual how shall I say, scale to the job that they're doing. They run at a very slow rate, 10 times, 1 times 10 to the third replications, maybe 15, 1.5 times 10 to the third. Very slow for an enzyme, actually. And it has a caboose on it. It's actually got 2 parts, and it basically can correct its own mistakes.